For 35 years, Dr. Carl Bassi has been on the faculty at UMSL College of Optometry, where he's a curator's distinguished teaching professor. His research and treatments aid patients, and I'm connecting with this, with macular degeneration, which my father had, Alzheimer's disease, which my father had, glaucoma, which my grandmother had, and progressive myopia. Yes, Carl is interested in eyeballs. But it turns out he's had a thing or two to do with golf balls. Some of you may know this, but he's worked on developing the triple track golf ball for Callaway. And it's had quite an effect in lowering scores of some major players. So, fun fact, take it away.
How about that? So I'm going to talk today about macular pigment. And the bottom line, the punchline for everything I'm going to talk about is macular pigment is more than just pigment in the macula. There's these same pigments that we see in the eye, they're found throughout the body. And we may be able to use our measurements within the eye to figure out what's going on with some of these I'm calling macular pigments, but they're really these other pigments that are there that are found throughout the body. Macular pigments were first described in 1780 by Bassi. He saw a yellow pigment within the eye of a cadaver eye, and it was then found. It's been worked on by a number of people, but it turns out this is not just a single pigment. There's a number of carotenoids that make up macular pigment, and that includes lutein, zeaxanthin, and mesozeaxanthin. These macular pigments they're not synthesized in the human body. They all absorb short wavelengths of light. There's a lot of studies now that show at least correlations between low macular pigment levels and an increased risk of macular degeneration. And again, these carotenoids that I'm going to talk about in the eye, they're found throughout the body. I first became interested in carotenoids exactly 20 years ago. I was approached by Dennis Gerhardt at Zia Vision in St. Louis. He was interested in macular pigment, quantifying how much pigment's there in the eye. Up until that point, the way you could quantify macular pigment, it required a lot of time and effort. You had to train patients or subjects, whoever was doing it. You had to train them. It took days. It took a long time to collect all the data. Dennis was working with a group at the University of Manchester, and we ended up being the first site in the United States to work on one of these early prototypes, and we greatly simplified the system to be able to collect data on these macular pigments. So we can actually quantify things, which is now developed into what's termed a quantify device that allows us to measure these macular pigments in both the clinical setting and also in the research setting. The other question that Dennis had for me was that there was these anecdotal reports that people who were getting supplemented with zeaxanthin, they reported that they had less glare sensitivity. And that actually made a whole lot of sense to me because right at the same time, this is about the time when LEDs were first starting to be used in car headlights. And you can see from the spectrum, there's a lot more blue light that's present in the LED lights than what was the normal incandescent lights. And it turns out that blue light, we're much, much more sensitive to glare. Well, it turns out the macular pigments, it turns out the macular pigments are absorbing right in that same position for the short wavelengths of light. So we ended up developing, again, this was something that was not easily quantifiable before. We developed a very simple device so we can measure glare sensitivity. It takes about 30 seconds to do. It's very reliable. It was shown to be people who had glare sensitivity, they required less light in order to have discomfort. We also used it in a supplementation study to show directly that those same patients who were getting an increase in macular pigment had a decrease in glare sensitivity. There's now been a number of studies that have gone on to find this same kind of relationship. I then began working with the Air Force. Chris Putnam was a research optometrist in the Air Force, and he came to my lab to do a PhD. He was interested in glare with pilots. You can imagine with a pilot, if there's a glare source that's very, very far away, that's going to project a very small image onto the retina, a very localized image onto the retina. It's not sort of this big glare source all the time, and maybe these localized things that are happening. So what we ended up developing was two devices. One device to be able to measure macular pigment within specific regions of the retina, and also go back and test in those same patients what was visual sensitivity in those same areas. We could look at contrast sensitivity with and without glare. So now we can look at a very local level to see how macular pigment affected sensitivity to glare. We had a couple of just basic findings. On the left was, first of all, there's big differences between individuals of what the shape of the distribution of how macular pigment is in the retina. Sometimes you have this sharp peak, sometimes it's a more broad peak. You can't, it's probably hard to see where you're sitting, but even absolute levels are very different. There's twice as much pigment in that center retina for the bottom one than there is for the top one. So there's these distributions and profiles, but yet in every single one of these patients, it turned out that at a local level, if you just looked at where you're actually measuring things from, there was going to be this decrease in glare sensitivity that was associated with this macular pigment level that was there. So we were able to show that again, just for the first time, to show this direct correlation of pigment and glare. We went on to do a number of other studies. We looked at light scatter, a number of other diseases within the eye that also affect central retina. And again, in all of these patients, what we end up seeing is changes in macular pigment levels are going to be either associated with things of decreasing light scatter, which is a good thing, or there's going to be these other changes that are happening with these areas that affect central retina are going to be associated with decreases in macular pigment levels. That wasn't what I came in here to talk about. I said that it's more than just the eye that's going on. And it turns out macular pigments do a lot more than just absorb blue light. They reduce phototoxic effects. This is interesting. I'm going to talk about this in just a second. Increased neural efficiency. They have anti-inflammatory properties. They have antioxidant properties. They can regulate apoptosis. There's a whole lot of things that these pigments are doing. There was a study that came out that showed that there was a significant correlation between macular pigment levels and those exact same pigments that are found in the brain. So we call it a macular pigment, but lutein and zeaxanthin are also found in the brain. And if you correlate how much macular pigment, zeaxanthin, and lutein there is, it correlates very strongly with those same carotenoids that are found within the brain. Related to that is there's been a number of studies from Renzi's and Hammond's lab that showed that there was an increased speed of processing and reaction times for patients who had high levels of macular pigment. So one of our questions is, can we see this again in just a very simple kind of clinical setting? Many of these studies involved a lot of very rigorous testing that took sometimes weeks to collect data. We have a system in our clinic that's called the CogniView. The CogniView is the only FDA-approved device for doing screening for cognitive deficits. The interesting thing about the CogniView is that it incorporates a number of tests, and if you look at these tests, many of these are vision related. So things like shape perception and motion perception, even when they're talking about shape memory or motion memory, these are still things that are associated with the visual system and what's going on with the visual system. The nice thing about the CogniView system is we can separate out motor speed versus what's going on with neural processing speed. So we just finished up this study. It actually just got accepted for publication, and I can talk about it here, but we looked in a group of patients where we directly compared macular pigment levels and how people performed on the cognitive view. And what we ended up seeing was there is a strong correlation between macular pigment and neural processing speed. People who had more macular pigment had faster neural processing speed. So I'm going to just jump ahead and just talk about a couple things. I just want to throw out, because again, I think the idea for this is think about collaborations. These certainly aren't, these other areas aren't my areas of expertise, although I know a lot about Alzheimer's. But these other areas I haven't had a lot to do with, but I just want to mention some of the other studies where we know carotenoids are associated with disease. With Alzheimer's disease, melanoma, the same pigment that we find in the eye, that zeaxanthin and lutein is also found in the skin. And it turns out that that may have the same kind of protective effects. Periodontal disease, heart disease, and there's a recent study with kidney disease. All of these seem to have these impacts of carotenoids with function in other areas of the body. So we think we have a tool. It's certainly important for the eye. It's absolutely important for the eye and think about what's going on. But we can use that tool maybe to get a window on some other things that are going on in other areas of the body. So I'm going to just stop there with that.
All right, thank you. Okay, I just want to say that for this particular Q&A, there's an incentive to ask your question at the microphone. Okay, even if you've already submitted one, if you're curious about what that means, come to the microphone. So who's got our first question? Yes.
Have you or has anybody used this tool to look at whether it's predictive of diseases like early stage Alzheimer's, neurodegenerative diseases?
So
Is it associated with it?
So it has not been looked at, to my knowledge, it's not been looked at for early stage Alzheimer's, but there is a group in England right now that's using supplementation in patients with Alzheimer's disease that looks like there's improved memory performance in those patients.
Thank you.
Next question, please.
This is a very naive question, but where are the carotenoids coming from? Is it all diet-based?
All diet-based.
Okay, so people that have increased carotenoids, they just have a different diet? Or is there some sort of processing in the body that retains them or something?
That's the more difficult question,
Yeah.
of course, and it turns out that sometimes it's strictly diet-related. Many times, if you just change diet, and the things that have these carotenoids is kale, corn, those are actually kind of the two big ones. And it turns out because of corn, it's also in egg yolks. The yellow in egg yolks has a lot of these same kinds of pigments. So sometimes people can improve it just with diet alone, but sometimes this supplementation seems like it helps. I will say this, I know there's been a couple of non-responders from people who get supplementation. There's got to be more than just getting it into the body. There's something else in terms of processing too.
One more question?
Really interesting talk. So it seems like zeaxanthin and lutein have really broad application in health, and you were talking about supplementation. But have any studies depleted those two nutrients and then seen what phenotypes present first to see which areas of physiology is most sensitive to these components?
Good question, and the answer is that what you the short answer is yes, that has been done in animal models. The only animals that have these macular pigments are primates. So this was a monkey model that was looked at, and what you see is if you deplete these macular pigments, if you do it for a long enough period of time, you actually start to see changes in central vision. That's what ends up happening.
Okay, thank you.
Great. All right, Paula, Catherine, and Randy. You heard me say earlier that Dr. Bassi developed the triple track golf ball. And you're going to get a sleeve of triple track golf balls that Dr. Bassi has signed. And I hope that if you are not a golfer, you're going to get this sleeve of balls to somebody who is. All right. So there you go. Little fun, little fun.